The last population of cells we'll discuss are the cranial neurocrest cells and the cardiac neurocrest. And remember that the cardiac crest is a subpopulation of the cranial crest. Most of the structures of the head and the face are derived from the cranial neurocrest cells, and these are labeled here in the green. And experiments have shown that cranial neurocrest cells are very heterogeneous in terms of their commitment. So that means that the cells are at different stages of commitment when they delaminate, and only about 10% of these cells are highly multipotent. So that does not mean that the other cells are unipotent, but rather that their fates are more restricted. So only about 10% of them are able to give rise to all the different types of derivatives that cranial neurocrest cells normally give rise to. And the rest of them give rise to a few or a subset of these derivatives, which include bone, cartilage, glia, neurons, melanocytes, and also muscle cells. The cranial neurocrest cells begin to migrate even before the fusion of the neural folds, and they migrate from regions anterior to rhombomer 8. Remember that rhombomeres are sections of the hindbrain or segments of the hindbrain. Now, these cells are going to migrate into the pharyngeal arches, and these cells that migrate through pharyngeal arches are going to give rise to many of the bones and cartilage of the face, and some of them even contribute to some glands like the thymus and the thyroid gland. In addition to that, there are cells that migrate more anteriorly, and those contribute to a region that's known as the frontonasal process, which forms most of the structures of the face. So these mesenchymal neurocrest cells contribute to many of the structures of the face, including the facial bone, cartilage and bone of the jaw, as well as cartilage of the larynx and trachea, bones of the middle ear, and although not shown here, these neurocrest cells also contribute to some of the ganglia of the face, so some of the cranial nerves that innervate the teeth and the jaw. In a previous video, you learned that cranial neurocrest cells use the process of collective migration to migrate as clusters, and this involves the low levels of expression of encadherin, the secretion of a coattractant C3A, and the process of contact inhibition of locomotion. In order for these cells to be kept as separate streams that are going to migrate into pharyngeal arches, which are pouches of endoderm, there have to be other processes at play. One of the factors that prevents these cells from mixing with each other is the presence of semaphorin and ephrins, which serve to repel the cells. These cells also have to be guided to the correct location, and this is achieved through a process that's known as a chase and run model. And basically this involves the secretion of a chemoattractant known as CDF1 by the ectodermal placodes, and these are thickenings of the ectoderm that are going to give rise to things like the lens of the eye, also hair, teeth, and these placodes secrete the chemoattractants, and the neurocrest cells contain the receptors CXCR4 for that chemoattractant. So they they follow the gradient of the chemoattractant. So this process of following after this increasing gradient of a chemoattractant is the chase aspect of this model. Once the neurocrest cells make contact with an ectodermal placode cells, the process of contact inhibition of locomotion will cause the placode cells to move away from the neurocrest cells, and this is the run. But these cells continue to secrete the chemoattractants so that the neurocrest cells still continue to chase after them and are thus able to move to the correct locations. The neurocrest not only contributes to bones of the face but also to bones of the skull, particularly the more anteriorly located bones, with the exception of the interparietal bone, which is more posterior. But in general, and this varies from species to species, the more anterior bones of the skull are provided or come from the neurocrest, whereas the most posterior bones come from the paraxial mesoderm. Perhaps one of the most important functions of the neurocrest is its role in regulating the growth of the brain, particularly the growth of the forebrain. In the chick embryo, it's been shown that when the cranial neurocrest cell is removed, the anterior portion of the brain, particularly the telencephalon, does not form properly. And this process can be rescued by adding beads that secrete FGF8 at the region of the anterior neuropore. Now, the neurocrest cells actually don't secrete FGF8, so their role is indirect. they secrete inhibitors of BMPs, and that allows the neural ridge or the anterior neural port to secrete FGF8s, which are needed for the brain to grow. Now, this explains some of the neural crest cell defects that are caused by teratogens that affect not only the facial structures, but also cognitive processes, because if the particular teratogen affects the cranial neural crest cells, it is likely that the growth of the brain will also be affected, because the cranial neural crest cells are so important for regulating brain growth. This FGF8 is not only secreted by the anterior neural port, and it's not only needed for the growth of the brain, but it's also secreted by the pharyngeal endoderm, and it serves as a chemoattractant for neural crest cells to be able to enter into the region of the heart and to form, among other things, the septum that is going to separate the aorta from the pulmonary artery. And because these cardiac neural crest cells are important for the formation of the septum, mutations in transcription factors that are required for either the survival or the proliferation of those cells lead to what is known as a persistent truncus arteriosus, which basically means that the septum does not form, and this is something that is seen in humans. In addition to contributing to structures of the heart, it's also important to know that the cardiac neural crest cells also contribute to cells of the immune system, because they contribute to the thymus gland, which is important for the function of the T cells.